Hello and welcome to the Natural Sciences at St. John's. My name is Eric Miske. I'm a Director of Studies and Supervisor at St. John's. I'm also the Herschel Smith Professor of Molecular Genetics here at the Department of Genetics, and I run a research team at the Gurdon Institute, both at the University of Cambridge. I'm here to tell you a little bit more about an amazing course within the natural sciences, biology of cells. This course introduces you to all of the fundamental concepts of modern biology. It starts off with the concept of a cell biology. Then it will tell you about the chemistry of life or biochemistry. You will get introduced to the fundamental concepts of genetics, which are important if you're interested, for example, in understanding evolution or how to identify human disease genes. This course ranges from the very molecular, at the level of molecular biology, towards the concepts of cells and how cells interact with each other, or cell communication. That gets you from the cell to tissue to organs. And finally, we'll learn about development. Let me just give you a little bit more of a background about this course. What will be different probably from most courses you've had at school is that there's a focus on the open remaining questions. Because despite the fact that there are tons of textbooks which already look quite thick about biology, there's a lot we do not understand. And it's very important to get to a level where you appreciate where our knowledge ends. It's also important to realize that all of this knowledge doesn't come out of thin air, but instead you will be introduced to the key technologies and the type of experiments that have been carried out to get to the understanding that we have today. This course consists both of lectures, and some of them are awesome. In addition, though, you're going to get your hands wet, so to speak, because there's a practical component to the course too. While, as you will see, this course will connect very easily to a number of different other courses within the first year, there are some key advantages of taking this course when you're thinking about next year. In fact, it is really essential for second-year courses such as biochemistry and molecular biology, cell and developmental biology, and plant and microbial sciences. Okay, what about these cells then? Well, we can think about cells in very different ways. They're physical entities, and we can think about the forces that act upon them. They are a bag of chemicals, and we are very keen to understand all the chemical reactions that are carried out within these cells. And amazingly, these cells are not created anew, but they have the ability to divide, and one cell makes two cells, makes four cells, and so forth. What's going on there, in particular with regards to the genetic material? Here's an amazing, very special cell, or if you look very closely, there are two cells. It's a human egg, and it is just about to be fertilized by a human sperm, which you might be able to make out in the needle here. So what's happening here? We've got this huge bag of an egg. We'd like to understand all the macromolecules within it. We'd like to understand what this membrane, the bag, is made out of. What is kept inside? What is kept out? What's the communication between the inside and the outside world? And of course, once this fertilization event has happened, this one cell will divide into many, many cells, and they will go on into forming an organism. How does this work? Again, all of this will be part of this specific course. At the very beginning, you will be introduced to different types of cells that we're used to seeing, eukaryotes and prokaryotes, and there might be a little bit of discussion of viruses as well. Once this concept of a cell is there, we're going to dive into the chemistry. The macromolecules play in a particular important role here. What's this DNA and RNA stuff, proteins, lipid, sugars, and so forth. My favorites are nucleic acids, and I want to introduce you to a very visual experience to nucleic acids that you can have here in Cambridge, although you might want to hurry up to do it very soon after arrival, or you want to wait till spring. Here's a pattern that depicts a piece of human DNA, and it's color-coded, which is a common thing that is done in illustrations. You see a blue stripe, one blue stripe, unfortunately, red, yellow, and green. And these 4 colors stand, of course, for the 4 nucleotides that make up our DNA. In fact, you can find them on a cycle path that goes from the university hospital, the so-called Edinburgh site, to one of the neighboring villages, Shelford. And in fact, if you write down the color scheme and translate it, if you will, into nucleotide or base sequences, you will see that this represents part of the human breast cancer 1 gene, a gene that is mutated in familial breast cancer. And this bike path was designed in recognition of the contribution that was made locally to discovering this gene and its role in heritable breast cancer. Once we get to this understanding of DNA as the molecule of heritability, we can fast forward to the very recent research on how we can alter it. You probably have all heard of the sort of technology of CRISPR and how it has the potential for a lot of medical applications in the clinic, how it is changing research in our laboratories, and also the possible impact on society, and that we really should all think about this a lot. Well, in this course, you will get to know how CRISPR actually works at the level of the chemistry of life, the biochemistry of CRISPR, as well as the implication for genetics at the level of an individual cell, an individual organism, a population of organisms, and, if you will, at the level of epidemiology. Another important aspect of chemistry of life is energy. How do organisms fight entropy? How can they keep an ordered state when everything else around us finds it very difficult to keep order, including the college room behind me? To do this, biological systems need to generate energy, and there are different ways they do this. And we'll learn the basic concepts of the chemistry going on there. And of course, it's not just chemistry, it's again physics and also the physicality of the building blocks of a cell. Here is a very bad cartoon, to be honest, of a mitochondrion. How is a mitochondrion built to provide the energy of life? Chemistry of life also goes a bit bigger. I already mentioned macromolecules and the DNA and proteins, lipids, and so forth. But many macromolecules assemble to larger complexes that are really the molecular machines that carry out chemical reactions, that can apply physical forces in biological systems. And to understand this, we really need to understand the mechanics of these machines. And we also need to understand the enzymology involved here. Again, chemistry and physics, very important here. Next, you're going to get an introduction to genetics. And genetics, as a pure science, is something that doesn't necessarily have to do with molecules. And you'll get this very classic introduction to genetics. This is important because it allows you to think very differently in an abstract way about heredity. However, this course doesn't leave it there, but you will also link it up to the molecules, in fact, the molecules that we already talked about. So while heredity involves passing on certain characteristics from one generation to another. And this could be, as one famous scientist once said, green cheese or DNA, because we do now know it's DNA. But in classic genetics, you will learn the concepts, including the math and the statistics required for dealing with heritability, and then link it up with the molecules that are involved and the molecular machines that are key to preserving this information encoded in the DNA. Let's go beyond the individual cell, because a lot of living systems that we're interested in, we realize they're made of more than one cell. This only works because cells communicate with each other. And in fact, even organisms that exist only on one cell often communicate organism to organism with each other, cell by cell. Here's an example that is maybe kind of small to read here, but it's a depiction of some basic concepts of iron metabolism. Iron that is ingested by your body is taken up in your intestine, and intestinal cells, they process this iron, they store it, then they can make it available to other cells in your body. There are other storage organs, for example, your liver. How is this controlled? This is where we move from the cell biology to cell communication, and we're touching on physiology. To understand physiology, we want to understand what's happening at the molecular level inside cells and how different cells, tissues, and organs link up within our body. Finally, and maybe you remember this egg that was about to be fertilized by a sperm in an earlier picture. To understand the physical form of organisms and to understand how this is linked up to the genetic material that encodes information for making this organism, how organisms adapt to the environment, how their physiology is controlled, and how over time organisms evolve to take on different forms, we need to understand development, because development gets us from this one fertilized egg to this organism with a whole physiology. How does this work? Again, this course provides the initial concepts of how our understanding of development has evolved over the last few hundred years. It will bring you right up to speed with modern technologies that examine development at a resolution that we've never seen before. Again, connecting molecules, cells that communicate with each other to the formation of a shape. And all of this is encompassed in the study of development. Okay, this is all, and it seems quite a lot, that is encompassed in this course. I can only recommend it to you most highly. And whatever you do, I just hope you have a blast at Cambridge. Thank you very much and goodbye.